you for the opportunity to be here. So my question is, where was the potassium at the origins of life? Potassium is the major intracellular cation in all living cells. And now how's this going to go to the next one? Come on. Okay. So I think this is the elephant in the room of research on the origins of life, that potassium is the major intracellular cation in all living cells, and we don't know where it came from. There are two possibilities for when the high intracellular potassium began. Maybe it began early and it was high in the earliest protocells. Maybe it came late. Intracellular potassium was low in the earliest protocells, but it became high at a later stage in life. And I say there, how? Or I could change it to how on earth? So cells need potassium for many functions from early in life's origins. Potassium's in the peptidyl transferase center in the ribosome, which makes it very old. Potassium's needed for protein translation. Potassium is crucial for the activity of numerous nearly universal key enzymes. This all comes from a paper by Dibrova and Mulkajanian and others. In terms of research on sodium versus potassium, Michael Dubina has a paper showing that potassium ions are more effective than sodium ions in salt-induced peptide formation. And here you can see his traces. And in potassium chloride, he's got an 8-mer oligo amino acid. And in sodium chloride, there's not much of anything as large as an 8-mer. So another piece of research from Paul Bracker's lab, linear dipeptides hydrolyze slower and cyclic dipeptides hydrolyze faster in potassium than in sodium. And so you end up with linear dipeptides and not cyclic dipeptides, which is what you want for life. So where was the potassium? As far as I know, there are 2 of us who've published on this, Mulkajanian and colleagues. Origin of first cells at terrestrial anoxic geothermal fields talks about potassium in this environment. My paper in Journal of Theoretical Biology is first, and it's actually still the best, the most detailed, about the possible origin of life between mica sheets, proposing that life emerged between mica sheets. So Mulkajanian, he says modern cells might have potassium higher than sodium because life originated in a habitat with higher potassium higher than sodium. This habitat is compatible with emissions of vapor-dominated zones of inland geothermal systems. The pre-cellular stages of evolution might have occurred in shallow ponds of condensed and cooled geothermal vapor that were lined with porous silicate materials enriched in potassium. So now I'll talk for quite a while about the possible origin of life between mica sheets. Mica is a non-swelling silicate clay mineral. And I have that little smiley face there to remind me to say that other clays like montmorillonite is much more common, and they swell and shrink as they wet and dry. And I don't think that something that swells and shrinks is a good habitat for the origin of life. Potassium ions hold mica sheets together. Potassium's purple in the flame photometer. That's why there's so much purple there, and the potassium ions are purple. So mica's anionic crystal lattice has the same 0.5 nanometer periodicity as extended single-stranded RNA. So you've got basically a template there. You can see the 0.5 nanometer scale bar up there between 2 potassiums where they're in between sheets. I picture sort of a solid state synthesis chamber with a crystal lattice ceiling and floor, beautiful for organizing polymerizations. And longer polymers are going to stick better. The little stuff's going to get washed away, and so stuff is going to grow. As Bruce showed us, mica is a good substrate for RNA synthesis at the origins of life. And these RNA polymers are just formed from things like AMP. It's not even anything as high energy as ATP, and certainly not those activated nucleotides that people were using on clay. And you can see there's a distinguished cast of authors down there on this paper. Mica is actually a good substrate for a lot of biomolecules. Lipids will, vesicles will fuse and form bilayers and multilayers. DNA will swim around on mica surfaces, proteins, laminin here. So living biomolecules are quite compatible with mica. And it's an atomically flat surface, which is nice for getting things started. So my hypothesis at the early stage, you can see the individual mica sheets, 50 of them in 50 nanometers. And the hypothesis is that there are a lot of compartments between these mica sheets, separated for giving isolation and interconnected for mixing, that polymers, little stringy things here, molecular aggregates, little blobs here, micelles and vesicles form on and between mica sheets in isolated niches and with occasions for interactions. And then at the edges of these mica sheets, here's a drawing and here's actual mica sheet. Whoops. Wow, I'm in a bind here. Okay, let's see, am I there? No, I'm here. Okay, so that there is the possibility of mechanical energy, and the mica sheets, in fact, will be doing mechanochemistry if there's any chemistry to be done at their edges. It's an energy source at the origins of life, and it's a very popular research topic in living cells now. And I'm not ignoring wet-dry cycles. So I'm not saying there's mechanochemistry without wet-dry cycles. There'll be wet-dry cycles at the edges of mica sheets and on their surfaces. I love this electron micrograph image of an air bubble in mica. So for the late stage hypothesis, I propose that all the essential components of living cells performed in different regions between mica sheets. Membranes were busy encapsulating all these components, forming vesicles. There's membranes forming. Vesicles fused and brought together different components into protocells. And eventually all the necessary components for life were encapsulated in a single structure in a living cell emerged. That cell probably died, and there were a lot more cells before life itself, actually, life before our ancestors of living cells emerged. So I talked about mechanochemical synthesis, and I had pictured big pieces of mica, and somebody raised the question, maybe little pieces of mica would work. So I have this diagram of the late stage hypothesis. I stretched it out to see what it would look like at 80 microns. This little, sorry. This little piece here is an 80 micron piece of mica, tiny piece of mica. I show here micaceous clay in a wet dry cycle and mica that was washed out of this micaceous clay. Where might micaceous clay have been? In clay containing hot springs. So mica's old, it's there at the Hadean. Micaceous clay is not as old as bulk mica because it takes a while for the mica to break down. So there won't be a lot of micaceous clay at life's origins. It's sort of like borate for those of you who are following the borate story. But there might have been a site with enough micaceous clay to provide potassium for life's origins. And finally, this is my proposal. Here you have Mulcajanian's terrestrial anoxic geothermal fields and Damer and Deemer's hot spring hypothesis. And all they need is micaceous clay, and you can get life between mica sheets with potassium in it. Potassium, intracellular potassium. I'm eager to interact with anybody who's interested in this subject. And so my email is hhansma@physics. Actually, it's just hhansma@ucsb.edu works, but at any rate, that's another one. So thank you, and I'm ready for questions.